From acute pain to chronic pain. He's going to be talking today on mechanisms governing the transition to chronic pain. Ted Price.
Thanks to the organizers for inviting me and putting on what's been a really great symposium today. I am going to talk about something that's slightly different than my title. I'm going to go where the data led me since I first submitted this title, and I'm actually going to talk probably a little bit more about mechanisms of neuropathic pain and how that helps us find new targets than about the transition from acute to chronic pain. But that'll still be a theme of what I talk about. So first of all, this is my lab group. My lab works as a collaboration with Greg Dusor. We've done all of our work together for many, many years now. Greg is right there. And I'm going to tell you guys a couple of different stories that have come out of my lab or that I hope will be coming out of my lab. Most of the data I'm going to show is unpublished. I'll start with a story from Jamie Moy, who's a PhD student that just finished, who's going to Michael Gold's lab to do a postdoc. Then I'll tell you about the work that Salim Magat and Pradipta Ray, who are both postdocs in the lab. Salim is on the biology side, Pradipta is on the computer science side. This has been a really nice collaboration between the two of them. Have been doing on chemotherapy neuropathy. And then I'll end up with somebody who's not in the picture, Paulino Barragan Iglesias, who's another postdoc in my lab who's been working on methylglyoxal and diabetic neuropathic pain. So this is a painting called a stab in the arm. And if you imagine that if you were stabbed in the arm just now, this is a pretty good example of what would happen from a neuroanatomical perspective, as well as your reflex reaction to that. The stab would cause your nociceptors that innervate the area to become active and start firing action potentials, sending a signal into the central nervous system, which enters at the level of the spinal cord, where the first synapse is, goes on up to the brain and tells you, number one, to get away from whatever is hurting you. So pain provides an absolutely vital protective function. But then over time, from that stabbing, you will continue to feel pain in that area, and that's coming from the inflammation and the tissue healing. And during that time, pain also serves a vital protective function because it teaches you to protect that injured area while it heals. So what happens in many people that develop chronic pain is that when that injury resolves, the pain does not resolve along with the injury. So we view this as chronic pain being a disease of the nervous system, a disease of neuronal plasticity. And one of our goals is to try to reverse that neuronal plasticity by identifying targets and then creating drugs that can target those mechanisms and potentially reverse them. So in this case, imagine instead that this person had a cervical spinal cord injury, maybe a disc herniation, and felt very much like they were being stabbed in the arm all the time, had a surgery to repair the disc herniation, and you can look at the structure of the spine, and it may look perfectly normal on imaging, but this person continues to feel pain as if they're being stabbed in the arm, and it can last for many, many years. This is a not uncommon situation at all for people that have chronic pain. So in that case, I would argue that what's happening is actually very similar, and this kind of builds on what Kyle talked about earlier. A key feature of how neuronal plasticity occurs in the nervous system is that neuronal plasticity drives phenotype changes in dorsal root ganglion neurons, in particular nociceptors, that causes them to transition from being in a normally quiescent state where they're not firing action potentials if they're not receiving any kind of stimulus, to firing action potentials what we call ectopically. So they're now not receiving a stimulus, but they are sending a signal as if a damaging stimuli is there. So they've transitioned from being silent to ectopically active. And in our view, that is really a key feature of underlying disease and probably a key feature of underlying disease, especially for neuropathic pain, which is what I'm going to try to focus on today. So our goal is to understand transduction mechanisms that cause acute pain, understand mechanisms that promote hypersensitivity and spontaneous activity during the recovery process, but really to focus on how this can transition to this. So this plasticity persisting after the healing has resolved, and now pain is clearly not protective and is a disease in and of itself, a disease that we argue is a disease of the nervous system, and I think most neuroscientists working in this area make that same argument every time they give talks. And so our ultimate goal is to try to discover these mechanisms, to eliminate that abnormal pain without interfering with this part, which is what we need to maneuver through the world on a day-to-day basis. So why do these neurons change their phenotype? Well, we know that that involves changes in gene expression. And many people have focused on the transcriptional step of this information transfer, so going from DNA to RNA. So I'm going to focus instead on translation regulation. This is obviously plays a critical role, but there are some reasons that we focus on translation regulation. One of them is that it wasn't until fairly recently that it was really widely appreciated that gene expression was highly regulated at the level of transcription and translation. We know now what the major steps regulating translation are. They're initiation, elongation, and termination. But the rate-limiting step and the most highly regulated step is the initiation step. And in the nervous system, translation regulation is particularly important because neurons are very, very large cells that have a nucleus that's found in the cell body, which in terms of the function of the neuron is actually the most boring part of the neuron. It's the axons and the dendrites that are really critically important, but transcription can't change in the dendrites and the axons because there is no nucleus in the dendrites and the axons. So translation can occur in any part of the neuron, and it can be controlled rapidly by signaling through neurotransmitter receptors that activate kinases that regulate the initiation step. This is reversible because these kinases can be turned on and off, and as I mentioned, there's a great degree of spatial control over this. So this is some electron micrographs from what I would argue is one of the most important papers in the neuroplasticity field. This is a paper that was published in 1981, I think, by Oswald Stewart, who is at University of California, Irvine, and is a luminary of the synaptic plasticity field. And the observation that he made was exceedingly simple. He was simply taking sections of hippocampus and doing electron microscopy and looking at where they saw ribosomes in these cells. So ribosomes do the action of protein synthesis. So the observation that he made, again, was very simple, and it was this. If you looked in the dendrites of these hippocampal neurons, which are huge, and looked at the places where you found ribosomes, you almost always found them in the same place, and that is at the base of dendritic spines. And the reason that's interesting is that these dendritic spines are where the synapses are. So that suggested that local translation at synapses was regulating synaptic plasticity, which I think most neuroscientists agree is an underlying neurophysiological principle that governs learning and memory. So in the now almost 40 years since he made that very simple observation, that there have been thousands of papers published on the role of local protein synthesis in synaptic plasticity, and we now have a pretty detailed understanding of how this works. And the basics are like this. At synapses, you find neurotransmitter receptors, which is not surprising because that's where neurotransmitters are released to act on those receptors. You find ribosomes at the base of these dendritic spines, but you not only find ribosomes, you find all the rest of the translational machinery. So you find mRNAs, you find mRNA binding proteins that move RNAs around in cells. You find the kinases that signal to regulate activity-dependent translation. And that signaling pathway boils down to three basic features. A kinase called MINC, which I'll talk about today, another kinase called mTOR, which I'll also talk about, and proteins that bind to the 5' end of the mRNA that regulate the initiation step. So translation can proceed without activation of these pathways, but it can be ramped up dramatically by turning these pathways on. And we know from the synaptic plasticity literature, but also from the cancer literature, that these signaling pathways play a fundamental role in synaptic plasticity and cellular plasticity that leads to oncogenesis, for instance. So I'm going to tell you guys today some stories about how we found that this also seems to play a key role in pain chronification. So when I first started my lab at University of Arizona, we were doing really simple experiments, just taking things that we knew caused pain in humans, exposing them to dorsal root ganglion neurons and seeing whether they increased translation, and importantly, seeing if they were increasing translation in the axons of those neurons. This is just one example of some images from a paper we published a long time ago. The green is the protein synthesis signal. Hopefully you can appreciate that IL-6 and nerve growth factor increase this really dramatically. They do it in the cell bodies, but they also do it in the axons, which you can see a little bit better over here. And this is blocked by the mTOR inhibitor rapamycin. So we wanted then to try to understand whether changes in these signaling pathways were involved in neuropathies. So we used a classic model called the spinal nerve ligation model that was developed by Jinmo Chung in the 1990s. And we simply looked in the injured, the uninjured DRG, and the sciatic nerve, and I'm going to focus on the sciatic nerve, for if these signaling pathways were turned on, if proteins that move RNAs around were increased, and if there was an increase in the overall translation machinery. And basically what we found is that all of those were dramatically increased. So mTOR and MINC signaling go up quite dramatically. Their downstream targets go up very dramatically, and proteins that move RNAs around in cells go up quite dramatically, as do things that are involved in RNA processing. So that's through Western blotting. We also used immunohistochemistry to localize these, and sure enough, they're found in the axons. For instance, you can hardly see any activated mTOR at all in axons in the naive state, but after injury, you see them decorating these axons. There are other things that are found here that are also positive for this. We now know what those cells are. Those are macrophages that infiltrate into the nerve after injury. So these pathways are turned on in two places, and we think that it drives neuropathic pain. That's in immune cells that come into the nerve, and also within the axons of the neurons themselves. So all of that kind of work culminated with this idea that the mTOR pathway and the ERK-MINC pathway leads to phosphorylation of factors that bind to the 5' end of mRNAs to dramatically enhance translation, and that increases the excitability of these cells, much like the signaling process drives synaptic plasticity in dendrites in the brain. So we wanted to get more detailed about this, and we started what's been a very productive collaboration with Jeff Mogul at McGill and Nahum Sonnenberg at McGill that's been led by our Katie Kortowski, who Luda actually talked about, who's a new professor in anesthesiology now at McGill. I did my postdoc at McGill, which is where I started working with most of these people. So the Sonnenberg lab developed this EIF4E phosphomutant mouse. So EIF4E is the cap binding protein on mRNAs, and it's only phosphorylated at a single site. And so we used this mouse that lacks phosphorylation of EIF4E to assess the role of phosphorylation of this protein in pain plasticity. And that phosphorylation event occurs downstream of MINC. So MINC is the kinase that phosphorylates this. So the idea here is to assess whether this plays a role in plasticity in the pain system, and discover whether this is a good target for manipulation of pain, because MINC can be targeted by drugs like circosporamide. So this paper's published now, so I'm not going to go into a lot of detail. This is the work Jamie did for her dissertation. I'm just going to show you a little bit of our work on intrinsic plasticity in DRG neurons here. What we were doing was exposing either wild type or EIF40 phosphomutant neurons to NGF. And you see NGF increases the excitability of wild type neurons, but it completely fails to do that in EIF40 phosphomutants. So this downstream signaling event is required for nerve growth factor induced changes in excitability. That's with the genetic model. Can we block it pharmacologically? This shows that we can. So we used circosporamide, which is actually a natural product that's being developed by Eli Lilly for cancer. And you can see that circosporamide very robustly blocks the effects of NGF as well. So we can block this either genetically or we can do it pharmacologically. So this is a slightly different kind of experiment that we're doing now. We're using these things that are called microelectrode arrays, where we can grow neurons on these and record action potentials from a large number of neurons at the same time. This really increases the throughput of what we can do. Here I'm showing you what happens when we record from these neurons over about 2 hours. Here's the baseline. These neurons are quiet, as you would expect for DRG neurons. If we expose them to IL-6, you can see that they rapidly change to not being quiet anymore. They fire action potentials. Even after we take the IL-6 away, they fire action potentials. In fact, they continue firing action potentials for about 24 hours. It's amazing what IL-6 does to them. But this is in the phosphomutants. You see that the response to IL-6 is much smaller, and they don't sustain firing. So again, NGF and IL-6 both require the signaling pathway to induce plasticity in DRG neurons. So we did a lot more work in that paper, but it's published, so I'm not going to go into that in any more detail. The question we had moving forward is what on earth is actually being translated that is changing the excitability of these cells? So to do this, I asked Salim Magat to develop a new technology or to take a technology that had been developed elsewhere and bring it into the pain field. So this took Salim about 2 years to get up and running. It was a really heroic effort, but it's worked out very nicely, as you'll see from the data I'm going to show you. So what Salim did was to use what's called translating ribosome affinity purification. So these mice have a GFP tag on a protein called L10A. L10A is a ribosomal protein that's only associated with translating ribosomes. The gene is built into the mice in such a way that it's only turned on in cell types that express Cre. So we used NAV 1.8 Cre, which is the promoter that's essentially only on in nociceptors. So this is a nociceptor-specific ribosome tag. And what this allows us to do is very neat. It allows us to express these tagged ribosomes only in a particular cell type, where we can then take that cell type out of the animal, purify the ribosomes, then take the RNAs that are associated with the ribosomes and sequence those RNAs, okay? So what it gives us is a cell type specific transcriptome, but it's actually a lot more detailed than that because it's not all of the RNAs, it's only the RNAs that are being translated. So it's a cell type specific translatome. So it turned out, and this is just showing you that this actually works. So here are the tagged ribosomes of GFP. Salim looked at a number of different markers. It's very, very specific for the NAV 1.8 population. We don't see expression ectopically in any other cell types, so we think it's a very, very clean technology, as you would expect, because NAV 1.8 is very specific for the nociceptor population. So we had to change the way that we do things in my lab because of this technology. So it turns out that the major hurdle to us getting this to work was getting enough material. So it turns out that to do the experiment, you need to pool all of the DRGs from 4 mice to get 1 biological replicate, and you have to dissect those DRGs very, very quickly to make it work. Otherwise, the ribosomes start to fall apart, and it just doesn't work. So we envisioned doing this with your traditional nerve injury models like SNI, but we just couldn't do it. So we had to switch to something where the neuropathy, or at least the thing that could be causing the neuropathy, is exposed to all sensory neurons. So we switched over to working on chemotherapy neuropathy. So what we did was to use kind of a traditional chemoneuropathy model, and that's paclitaxel given at a cumulative dose of 16 mg per kg over 4 injections, so 4 mg per kg every other day for a total of 4 injections. And then we took tissues on day 10 when the neuropathy was fully established, and took the total transcriptome from the DRG and also the translatome, or the mRNAs that are associated with ribosomes. And what you see is that the transcriptome actually doesn't change very much from paclitaxel treatment. We only see about 60 genes upregulated, 20 downregulated. But the translatome changes to a larger degree, and if you look at whether these overlap, they don't overlap at all. So the transcriptional changes don't really seem to be coupled to the translational changes, which we would have predicted based on what we know about translation regulation anyway. If you look at the things that are changing in the translatome, we find, number one, that mitochondrial function is altered, which is actually very well known for chemoneuropathy. But we found several other things that we thought were very interesting and led us to some new mechanistic studies about chemoneuropathy. So that's what I'm going to tell you about now. So it was very interesting to us that among all the genes that were upregulated, many of them were related to mTOR signaling. So we wanted to try to understand, number one, if we could validate this, and number two, why is it happening? What's causing the mTOR signaling to increase? So first of all, we see with paclitaxel treatment in the DRGs that EIF4E phosphorylation is increased, 4-EBP phosphorylation, which is a direct target for mTOR, is increased, and total mTOR is increased, so the pathway is turned on very strongly. And second of all, we had these mice available already that I told you about, so we wanted to assess whether they developed chemoneuropathy or not. So we looked at wild type and EIF4E phosphomutant mice, and what we saw was that the mechanical hypersensitivity induced by paclitaxel was decreased in the phosphomutants. Heat hypersensitivity was very strongly decreased in the phosphomutants. So here's in the wild type, you see at several different temperatures they develop thermal hypersensitivity. There's very little development, or if any at all, of thermal hypersensitivity in the EIF4E phosphomutants. And finally, we use condition place preference with retigabine as the conditioning factor. I'm not going to go into the details of this experiment because it's kind of complicated, but those of you that are familiar with this idea probably get the idea. In the wild type mice, we got very strong conditioning to retigabine, suggesting that there's spontaneous pain induced by the chemoneuropathy. But in the EIF4E phosphomutants, there's no conditioning, which suggests that they don't experience spontaneous pain, consistent with the lack of mechanical hypersensitivity and thermal hypersensitivity. So this would suggest that MINC signaling could be driving chemotherapy neuropathy. So to test that hypothesis, we partnered up with a pharma company called Effector Therapeutics. They're in San Diego. They're developing the compound that I'm showing you here, EFT508, for cancer. This compound is a very specific and extraordinarily potent MINC inhibitor. It's in phase 2 clinical trials right now, so it's pretty rapidly moving towards the clinic. So our hypothesis is that this should inhibit MINC signaling in DRG neurons, number one, and number two, it should reduce chemotherapy neuropathic pain. So this shows you that the compound works, so it blocks EIF4E phosphorylation very effectively in dorsal root ganglion neurons. It doesn't seem to turn on feedback signaling, which is very common when you mess around with the mTOR pathway, so we like that it doesn't do that. And here's the behavioral data. If you give EFT508 at 10 mg per kg, it leads to a very rapid and fairly long-lasting reduction of mechanical hypersensitivity. It comes back after about 24 hours of a single dose, but if you give multiple doses, you continue to see relief of pain, suggesting that there's no tolerance induced when you give this drug. So we're excited about the concept that MINC inhibitors might be able to be used for a wide variety of neuropathic pain, including potentially chemotherapy neuropathy. So one of the big questions we had is why is this happening? What's engaging the mTOR pathway in chemotherapy neuropathy? So our original idea was that it must be mitochondrial related, because mitochondria are very well known to play a role in chemoneuropathy, but we actually don't think that's the case. So if we looked at our data more closely, what we noted was that a complex that sits on the surface of the lysosome that is called the regulator complex, which is a requisite for amino acid-induced activation of mTOR, is all extraordinarily strongly increased by the paclitaxel treatment. So our current hypothesis, and I can just show you a little bit of the data for RAG-A, which is a key part of that complex, is that this lysosomal deregulation or dysregulation is actually maybe the driver of chemotherapy neuropathy, and that the mitochondrial dysfunction actually occurs downstream of that. So I don't have any evidence to support that right now, but this is the hypothesis that Salim is now working on. And some of you that are aware of the literature for chemotherapy neuropathy may know that while there is clearly mitochondrial dysfunction, the clinical trials that have been done with things that should modulate mitochondrial dysfunction have not shown very strong effects. So we think that that may actually be a kind of a bystander effect of this lysosomal dysregulation that happens, and that what we really need to do is to target this lysosomal dysregulation. So that's a hypothesis. We're actively working on that. So this is just to summarize what I've told you so far about that we think that MINC plays a really key role via EIF4E phosphorylation. So we're still trying to understand the other targets that are regulated here, and obviously we don't know how lysosomal dysregulation leads to changes in neuronal excitability, but we're trying to figure that out. So I'm going to end by talking briefly about methylglyoxal and diabetic neuropathy. So this is a project that Paulino, Braga, and Iglesias has been working on. We got started on this because of this paper that was published in Nature Medicine now several years ago. And essentially what this German group found was that in patients that had painful diabetic neuropathy, but not non-painful diabetic neuropathy, one of the distinguishing factors was a high concentration of methylglyoxal in their blood, in their serum. So they demonstrate this very nicely in the paper. They show that methylglyoxal induces hyperexcitability, but the mechanism through which methylglyoxal acts has not really been ironed out. And one issue related to this is that methylglyoxal concentrations in blood of diabetics with neuropathic pain are about 1 micromolar, but a number of studies have used very high concentrations of methylglyoxal to show that it's a TRPA1 agonist. So there's an idea that methylglyoxal acts via TRPA1, but the pharmacologist in me sees these concentration response curves and says this doesn't really jive with the clinical story. So we wanted to try to understand at the pharmacologically relevant concentrations of methylglyoxal what is happening in DRG neurons. So this was the study that Paulino took on, but I want to just mention that we had one other piece of information that came from Bruce Hammock's lab at University of California, and that was that the integrated stress response is turned on in DRG neurons by diabetic neuropathy. So the integrated stress response is essentially a signaling pathway that regulates translation. But it regulates translation in a slightly different way. Rather than turning up translation, it turns down translation. And it does that by phosphorylating a protein called EIF2 alpha. But what's really interesting about this is that while it shuts down translation of canonical open reading frames, it turns on translation of what is known as an upstream open reading frame. So let me explain that quickly. So the main open reading frame of mRNAs is started by the start codon, okay, which is surrounded by a Kozak sequence. But in many mRNAs, there are alternative start codons that maybe don't have a Kozak sequence around them that are found in the 5' untranslated region. And some of those can encode peptides that also have a stop codon, or sometimes they can just read through into the main open reading frame. So when EIF2 alpha is increased, as it is in the nerves of animals with diabetic neuropathy, that leads to decreased translation of the main open reading frame, but enhanced translation of the upstream open reading frame. So it leads to an entirely different type of translation regulation. So we wanted to see if maybe methylglyoxal was doing this. So the first thing Paulino had to do is just figure out whether methylglyoxal causes pain in our hands, and it does. So here's the dose response. We find that very, very low doses of methylglyoxal, this is very consistent with what was found in the German paper, cause mechanical hypersensitivity. These low doses of methylglyoxal-induced mechanical hypersensitivity are not blocked by TRPA1 antagonists. So this is a well-known TRPA1 antagonist given at an absolutely monstrous dose. But they are blocked by general translation inhibitors. So we can block them with a ribosome inhibitor, which blocks canonical open reading frame translation and upstream open reading frame translation. So does methylglyoxal induce activation of the integrated stress response in DRG neurons? It does, and it does so at very low concentrations, so about 400 or 500 nanomolar, so right about at the threshold for what you see in patients with painful diabetic neuropathy. BIP is a MRA that is translated via one of these upstream open reading frames when the integrated stress response is turned on. PERK is a kinase that phosphorylates the EIF2 alpha. You can see it's activated, and the EIF2 alpha itself is phosphorylated. So we know that EIF4E phosphorylation regulates canonical translation, so we asked if methylglyoxal-induced pain is altered in the phosphomutants that I just showed you, all that work on paclitaxel, et cetera. Well, now we see nothing whatsoever, so methylglyoxal induces very robust pain in these mice. And it doesn't influence this pathway, so completely different type of pathway than what you see with paclitaxel, for instance. We also see that methylglyoxal reduces overall translation, so this is just a assay to do that. In red, you can see the puromycin incorporation. This is called a sunset assay. It's completely blocked by HHT, which is the positive control, which is a ribosome inhibitor, and it's inhibited fairly strongly by methylglyoxal, but obviously some translation is still happening. This is probably mediated by upstream open reading frames. And does methylglyoxal induce a change in excitability in DRG neurons? We use calcium imaging to address that. And you can see here that if we expose these neurons to methylglyoxal, we can enhance their excitability. So the final question then is, if we block the integrated stress response, can we block methylglyoxal-induced pain? So it so happens that there's an inhibitor of the integrated stress response out there called ISRIB. So Paulino used this molecule to do that, and it nearly completely blocks methylglyoxal-induced pain. So it works if we give it locally. So the methylglyoxal is given in the paw and preceded by ISRIB injection. It works if we give it locally. And if we give a really high dose of methylglyoxal that induces a really long-lasting pain state and give animals a dose of ISRIB, it also reverses their hypersensitivity. And it blocks EIF2 alpha phosphorylation in response to methylglyoxal. So all of this leads us to a couple of conclusions about how different types of neuropathic pain are regulated. So one thing that's interesting is all of these types of neuropathic pain, trauma-induced, chemotherapy-induced, or methylglyoxal-induced, all lead to the same phenotype. They cause thermal and mechanical hypersensitivity and spontaneous pain. I didn't show you the spontaneous pain data for methylglyoxal, but they do. They all rely on a particular type of mechanism, which is translation regulation. However, the underlying mechanism that's driving it is completely different. So a big point of today is precision mechanism. And I think what this is teaching us is that you have to really get into the nitty-gritty of the details. And in our case, we needed some pretty advanced genetic technologies to be able to do that. But now that we've done it, I think we have a better idea about what kind of mechanisms we might want to use for very specific types of neuropathic pain. So for instance, for chemotherapy neuropathy, we're extraordinarily excited about this MINC inhibitor from Effector that's in phase 2 clinical trials, that we could potentially start a chemotherapy neuropathy trial relatively soon. And the other neat thing about this is that not only does it work to prevent development of chemoneuropathy, but it works to reverse chemoneuropathy. But for methylglyoxal-induced pain, which we think might be tied to diabetic neuropathy, we would want a completely different kind of compound, and we're also very excited about this ISRIB molecule. So this one is not in phase 2 clinical trials, but it's a drug-like molecule. It's undergoing clinical development. It may get to phase 1, and we could be there at some point where we could use a molecule like this to potentially manipulate diabetic neuropathic pain. So I'll stop there and just want to again point out the really talented people in my lab that have done this work, especially Salim, Paulino, Jamie, and Pradipta on the computational side, analyzing all the RNA sequencing data. And all of this was initially funded by the Rita Allen Foundation and is now funded by NIH. So thank you very much for your attention.
That's a nice talk. I have a question about methylglyoxal. How is it made? Is it a byproduct? Is it specific to? So sorry, go ahead.
Yeah, so it's an inglycation product. It's produced as a result of high glucose. It's highly reactive, but the exact cellular targets are not very well known. We think it produces additional reactive oxygen and potentially reactive nitrogen, but we don't have a lot of evidence for that. There is, at least in our models, there's evidence for that in the literature, and that's kind of our active hypothesis.
Have you looked at blocking, say, Ross species or anything like that?
We have used a compound that we know blocks reactive nitrogen, and that works very efficaciously to block methylglyoxal-induced pain. I think that compound likely also decomposes reactive oxygen, but the information we have on reactive nitrogen is much better.
Thank you, Ted. I'm curious on your views on, I guess I'll call it off-target effects. That is, to what degree do you think we'll be able to control tissue or cell-specific inhibition of translation? And I guess sort of on the other side of the coin, to what degree do you think that the inhibition goes beyond the target of interest here?
So I guess the easiest way to answer that question is that the last really decade of work in translation regulation has led to a fundamental change in the way that we think about these pathways. So the idea not too terribly long ago was, you inhibit the mTOR pathway and you shut off translation altogether, and it's going to make people very sick. But many people take rapamycin. It's a strong immune suppressant, right? And I don't think that's really a good idea for your average everyday chronic pain patient, although there may be scenarios where it is. But MYC inhibitors, those have gone through phase 1, and they're safe because it's in phase 2. And I think that what we're really trying to do is to block activity-dependent changes in gene expression, and that if you do like a sunset assay or any of these measures of protein synthesis with MYC inhibitors, you don't see any change in bulk translation. What you do see is there's a shift in the mRNAs that are associated with the ribosome. So there's a pool that's kind of constitutively translated, and then if you stimulate the pathway, it shifts over to another pool of mRNAs, and then if you block the MYC pathway, you shift back to the constitutively translated pool. And our idea is that the RNAs that sit in the activity-dependent pool are plasticity proteins, and in fact, we know what some of them are, and they are. It's like ARC, activity-regulated cytoskeletal protein, which plays a role in dendritic remodeling. We don't know what it does in axons, but my guess is it does something very similar. CFOS, which is a strong regulator of transcription, CREB, which regulates transcription, a number of scaffolding proteins that are involved in regulation of ion channels. So that's really how we're trying to move ahead with that. So I think it's always a concern, especially with kinase inhibitors, that they're going to have some kind of side effect that is going to derail them. But you know, in the case of MYC inhibitors, that hasn't been found yet.
Great talk. I'm really interested in your studies in diabetic peripheral neuropathy, because not all patients develop it. And so when it's painful, we've been trying to look at different genetic indicators of that, predictors of that. How are you looking at that in future experiments in differentiating the mechanism that you're looking at right now in the face of that genetic background?
I'm not aware of any studies that have made any clear connection between genetic background and methylglyoxal levels. That would be interesting if they existed. There's a lot of controversy around methylglyoxal, so you know, I don't want everybody to leave today thinking methylglyoxal causes diabetic neuropathic pain, because other studies have not replicated that. There's some that have and others that have not. That it may be because of genetics. So, you know, there may have been cohorts that have a predisposition to higher levels of methylglyoxal with hyperglycemia, and that's driving it in some populations. So I think it's a great question, and it's not something can really answer right now. Something that is interesting is that a variety of other neuropathies have now been associated with high methylglyoxal levels. So there was a really interesting paper that just came out about low back pain, showing very striking increases in methylglyoxal around herniated discs. So it's possible that methylglyoxal is a potential driver of neuropathic pain and a variety of different types of neuropathic pain.
By way of closing, let me just say this, in my mind, was an absolutely fascinating symposium that was very wide-ranging and touched on many different things related to pain, from molecular biology all the way to psychosocial interventions. And you know, I'm coming away from this with the impression that there's a lot of work to be done at all sorts of levels, from the basic to the translational. And I want to thank all the speakers for coming here and giving us very inspiring talks, and the audience for showing up. Thank you.